Hello dear students, in our last lecture, we learned about the genetic material and discovery of genetic material. Now in this lecture, we will discuss structure of DNA. Correct structure of DNA was worked out by James Watson and Francis Crick in 1953. Their double helical model of DNA was based on two important points. Number one is Chargaff's rule of base pairing and number two X-ray diffraction pattern of DNA. वॉटसन एंड क्रिक को अपने डीएनए के मॉडल को सही तौर पर बनाने और पेश करने में इन दोनों पॉइंट्स की बहुत मदद मिली. अब एक के बाद एक हम देखेंगे कि ये दोनों पॉइंट्स किस तरह से डीएनए के मॉडल को पेश करने में इम्पॉर्टेंट साबित हुए. Erwin Chertoff formulated generalization about the DNA structure. These are called as Chertoff's rule and are summarized as purines and pyrimidines are always in equal amount. As we know that DNA is made up of nucleotides and these are building blocks or building units of DNA which are composed of phosphate group, deoxyribose sugar and nitrogenous bases. Purines and pyrimidines are always in equal amount, that is A plus T is equal to G plus C, and amount of adenine is equal to amount of thymine. That means A is equal to T and G is equal to C. And base ratio for A plus T is always constant for a given species. Now the second important point is X-ray crystallography. It is a technique for determining structure of large molecules. Maurice Wilkins and Rosalind Franklin took X-ray diffraction pictures of crystalline DNA, and with the help of this, Watson and Crick proposed their double helical model of DNA. Now students, Watson and Crick along with Wilkins received a Nobel Prize in 1962 for double helical model of DNA. This model has the following important points. Students, structure of DNA is not given in your board textbook. That's why I am explaining it in brief, as it is not included in your board syllabus, but to understand DNA replication, you must know structure of DNA. This model of Watson and Crick shows following important features. Number 1 point, two polynucleotide chains or strands, they form the backbone of DNA strand. 2. Polynucleotide chains, they have a specific polarity. They are anti-parallel to each other. These bases are complementary, and adjacent nitrogenous bases, they are joined by two different important joinings or bonds which are called as glycosidic bond and phosphodiester bond. And two nitrogenous bases, they are always joined together in the DNA double helical model by hydrogen bonds. Every polynucleotide chain or strand of DNA is made up of small building units which are called as nucleotides. Each nucleotide contains three important structures. Number 1, deoxyribose sugar, nitrogenous base and phosphoric acid. Now students, nitrogenous bases are of two types, as I have mentioned it earlier, purines and pyrimidines. Purines are of two types, adenine and guanine, and pyrimidines are of two types, thymine and cytosine. Now these nitrogenous bases, purines and pyrimidines, purines are large size nitrogenous compound containing 9-member double ring. They contains nitrogen atoms at 1, 3, 7 and 9th position. The name purine is small but structure is double ringed structure, and the name pyrimidine is large name but it has these nitrogenous bases have a small size nitrogen containing biomolecules. These are single ring compound. They contain two nitrogen atoms at 1 and 3 position. See students, polarity in DNA strand is very important. You have to understand it very carefully. Each DNA strand have a specific polarity which is 5' to 3' in direction. यानी DNA की दोनों पट्टियाँ आपस में एक सिम्त या डिस्टिंक्ट डायरेक्शन को जाहिर करती हैं, जिसे हम पोलैरिटी कहते हैं. One end of the DNA strand has a free phosphate group at 5th position of the sugar, hence it is called as 5' end, while the other end of the strand, sugar has a free hydroxyl group, hence it is called as 3' end. Base pairs are joined by hydrogen bonds. Two DNA strands are anti-parallel and complementary to each other. The two strands are parallel but oriented in opposite direction. That is, one strand with 3' to 5' direction and other strand is from 5' to 3' direction. The two DNA strands are not coiled upon each other, but the duplex is coiled upon itself. Coiling is plectonymic. That means two strands cannot be separated without completely unwinding them. Diameter of DNA strand is 20 Armstrong unit and 1 turn of spiral has a distance of 34 Armstrong unit. One spiral turn has 10 nucleotides, so the average distance between adjacent nucleotide is 3.4 Armstrong unit. Nitrogenous bases are attached to carbon number 1 of deoxyribose sugar with glycosidic bond, and the bond between two adjacent nucleotides of two adjacent sugar molecules at 3' and 5' position with phosphate group is called as phosphodiester bond. The two strands of DNA are held together by hydrogen bonds between their nitrogenous bases. Adenine and thymine joins with two hydrogen bonds, that is A double bond T. And between guanine and cytosine there are three hydrogen bonds, that is G triple bond C. Hence, the bond between guanine and cytosine is very strong as compared to adenine and thymine. See students, as I have earlier mentioned, the DNA structure is not given in your textbook, but you cannot understand DNA replication without knowing structure of DNA. I hope structure is briefly explained to you. Kindly go through the topic very thoroughly, as it is very important topic and not a single part from this topic is deleted by the board this year. Thank you very much.